Hello, thank you very much. So it's almost lunch, so we are all tired, so I'm going to avoid deep learning and instead try to concentrate on one simple idea. And I'm going to talk about plasmid identification. Why are we interested and what are plasmids? Just a reminder, because almost everybody in this room probably knows this. So plasmids are small circular DNAs. The DNA molecules, they are located typically in bacteria. They carry genes that come from selective advantage, for example, antibiotic resistance genes, so that's why they are interesting. And they facilitate horizontal transfer between individuals, and perhaps most important thing for us now is that they replicate independently of chromosomal DNA. So that means that there are observable differences between chromosome and plasmid sequences, and therefore we can try to identify plasmid sequences just by looking at sequences. So the problem that we are addressing, and that is like traditional problem that is solved, we have a bacterial genome assembled from a bacterial isolate. We try to identify which contigs correspond to chromosomes and which contigs correspond to plasmids. Some of you know, is it a difficult task? Is it not? Well, it depends. It depends how good is your assembly. If you are working with the long reads, then typically you get very good assembly, you get the long contigs, and there is no problem identifying whether this is a plasmid or chromosome. If you are working with short read assemblies, you get short contigs, and then that's where the difficulty lies. So in this talk, I'm going to concentrate on contig classification for short read assemblies that has many short contigs. So the standard approaches are based on machine learning, and they can be split roughly into two different categories. One is using only sequence. One is the category where you use only sequence features. So I have a couple of tools listed there. Basically, the features like K-MEC composition, GC content, coverage by sequences reads important. And the other one is basically, why would you not give to your learner how E. coli genome generally looks like? So you want to add some kind of homology features or something similar. So there are, again, like plethora of tools that do that. And basically, they typically add many features that either indicate some similarity or occurrence of a database hit or a specific hit of specific genes or protein family or something similar. So I'm not going into more detailed explanation of these tools. Okay, so what's the problem with how these homology features are added? Well, first of all, some sequences may play a dual role. So, for example, there can be some sequences that in one context, in one bacterial isolate, they will play a role. They will be part of plasmid. In other one, they will be part of chromosome. So depending on which isolate you look at, the chromosome or plasmid is a correct answer for the same sequence. So that might cause some problems. Second, reliance on homology features biases results to things that are already known. If you are looking at, I don't know, if you take and do a homology search to PLSDB and based everything on these matches, then things that are not in PLSDB will cause you trouble. And you will not even probably indicate that because there is a high likelihood that everything in your testing set is already in PLSDB. And the last thing is technical. So if you take and I was saying, you say presence or absence of protein family or things like that as a feature, right? But if you add like many features like that, we have seen this in a couple of presentations already. It's going to make machine learning complicated in some cases. At least for some models, this will likely lead to overfitting. And there are many ways how you can avoid it, but you have to work for it. Okay, so instead we are trying to take a step back and trying to take a much simpler approach to this. So in our work, we try to introduce some kind of text that will sort of have something to do with the fact whether something is a chromosome or a plasmid. And we try two different types of text. The first one is a very, very easy one. Basically, you take a PFAM and you look at the occurrence of PFAM families. The second one is a little bit more complicated. So basically, we take, let's say, we take a couple of bacteria isolates. We build a pangenome out of them. So that's what's on the right over there. And then you basically take the vertices of the pangenome graph, which basically represent the sequences that tend to sort of be included or excluded as a whole, not as a whole pieces from the thing. So they also make good tags, and they are otherwise not informed in any way. So you don't than the sequence to finding them. So so you take the vertices from the graph, and you see whether they occur in your sequences or not. So we have the tags. Now we need to assign some kind of score to the tags, and we just take a very simple logit score based score. Basically, you take each type of the tag and you compare at what frequencies they occur in plasmids and in what frequencies they occur overall, and you do similar, like a very simple logit score, similar to how we build, like, let's say, blossom matrices or things like that. So basically you do that. Then positive score will indicate that, yeah, this is more likely to occur in plasmids. 0 will indicate, or close to 0 will indicate, oh, okay, this is some feature that doesn't really have to do anything with plasmidness or not. And the negative score will give you a negative information. So we just and these scores, because they are built this way, they are simple additive scores. So you can take all the tags that occur in your sequence and simply sum them up and get the final score. So that will be our feature, and we will just use this one feature. Actually, we have one for plasmid and one for chromosome for technical reasons, but we will add 2 features to our model and see how it does. And so this is just a table that summarizes. So we have our, so this indicates, so if you look at the second column here, that is entitled DB, it says, does this tool use a database of some kind or not? So there are a couple of tools that don't use any database, just use sequence features. Our tool, Plasgraph2, also only uses sequence features. And we only concentrate for simplicity on F1 scores. And you can see that that when you add a PFAM or these other tags to the Plasgraph2, you increase the accuracy, the F1 score of in the evaluation significantly. So this actually works. This on this particular testing set, this is the Plasgraph with the PFAM and the other tags, other type of tags, is actually the best performing tools. So, and I think that's all. So the GitHub repository for Plasma is going to be updated in the next few days for the homology features. So it's not there yet. And I would like to acknowledge, especially the group at Simon Fraser University, where we started working on Plasgraph, and yeah, that's it. Thank you.
Thank you very much. We have time for a few questions.
Hi, great talk. I was wondering how much of the plasmid do you reconstruct, especially in your benchmarks, and how do you delineate, parts of the plasmid integrating into the chromosome versus, just the other parts of the plasmid?
Okay, so our goal was not to reconstruct the plasmids. Our goal was to take the assembly that has already done somebody else, that is already done by somebody else, and classify the contigs. And in fact, if you look at here, we are actually one of the advantages of our tool is that we can actually work with really short contigs. So we can classify contigs that are 100 base pairs long pretty accurately. And this is not done alone. This is done by utilizing the information from neighboring contigs that are sort of joined in an assembly graph. So we are not reconstructing plasmids per se. We are just taking the individual contigs and trying to classify them. Is this a plasmid or is this a chromosome?
So these escapes are, is that correct?
No. So these are isolates.
Okay, have you tried it on metagenomes?
So for metagenomes, Plasgraph2 doesn't work on metagenomes. There is a specific technical reason why that is. And so we cannot. So we didn't try this because we were adding these features to Plasgraph2. But the features themselves, there is no reason why they shouldn't work.
And then in a metagenomics context, quite commonly, well, I think most commonly used at the minute is GenomeAd. Have you tried comparing to that?
No, since we didn't work on metagenomes.
Well, that would work in the genomic context. So you could compare it. Anyway, okay, thanks.
Okay, well then, thank you again.